here to present her work over the past 5 years. And please also help yourself to get some food in the back. Maybe you didn't see it when you entered the room. And so we are waiting for another committee member to join remotely, but I think we should start on time without further delay. Wei joined our Department of Statistics at UCLA 1 year after I did. So she was my very first PhD student. I joined in 2013 and she joined in 2014. And I always felt very fortunate to have her as my student because I felt like her PhD career was kind of aligned with my junior faculty career. Like we tried so many things together. Without her, myself couldn't achieve what I have right now. So that's why I think it's a very big honor for me to be her advisor and also to introduce her here before her talk. So what she did in her PhD was to develop several methods for analyzing RNA sequencing data, which is a very important data type that has been widely used over the past 10 years in the biomedical sciences. So many people generate those data, analyze this data to make discoveries. But there are a lot of computational challenges there, so she developed very important tools. So let's welcome her to give this talk, and after she finishes the talk, we will first ask our audience to ask your questions, and then after you finish the questions, we will close the door to let the committee discuss her defense. So that's the plan. Okay, so welcome, Wei.
Thank you very much for the introduction, and I want to thank you all for coming to my defense. And a major focus of my doctoral research is to develop statistical methods for uncovering hidden information from large-scale genomic data sets. And in this talk, I want to introduce several statistical methods for us to analyze both bulk and single-cell RNA sequencing data. The RNA sequencing technologies have become the standard tool to analyze the presence and quantity of RNA molecules in biological samples. And the sequencing data allows us to address biological and statistical questions at different levels. In the first part of this talk, I will introduce two statistical methods we developed to improve the transcript-level analysis of bulk RNA sequencing data, which means that the RNA molecules are taken from many cells in the bulk tissue and merged together before the sequencing step. In the second part of this talk, I will introduce several methods for analyzing single-cell RNA sequencing data, which means that the RNA molecules are taken from isolated individual cells. So first of all, why does the sequencing data become so popular and important in genomic studies? The year of 2018 marks the 60th anniversary of central dogma, which states that DNA makes RNA makes proteins. And in central dogma, the transcription process refers to the synthesis of RNAs based on the DNA templates, and the translation process refers to the synthesis of proteins based on the messenger RNAs. In the transcription process, multiple exons of the same gene are copied into the RNA molecules, while the introns are removed or spliced out. So the central dogma explains how the genetic information flows from the DNAs to RNAs and then to proteins. However, in order to better understand how cells with the same set of DNAs can develop into tissues and cell types of different functions, we need to investigate more complex regulatory mechanisms. And 20 years after the publication of central dogma, people discovered that there's a process called alternative splicing, in which multiple exons of the same gene may be either included into or excluded from the RNA molecules. Let's take this 4-exon gene as an example. Its second exon is either included into the RNA molecules or spliced out, leading to two different RNA variants, and we call them isoforms. Different isoforms of the same gene may exist in different quantities, and in the translation process, it may produce proteins of different functions. The mechanism of alternative splicing contributes to the genomic diversity of different tissue and cell types. And this is a very important process in eukaryotes. While on the other hand, abnormal splicing may lead to genetic diseases. As an example, we suppose the third exon of this gene must be included in the RNA molecules in order to produce functional proteins in the normal condition. And in contrast, The abnormal splicing out of this exon may lead to non-functional proteins in disease conditions. As an example, the spinal muscular atrophy is the leading cause of infant mortality. And in these atrophied muscle cells, the abnormal splicing of a gene called SMN can lead to truncated proteins. And without a proper level of the SMN protein, the muscle neurons will be lost and they cannot receive correct signals from the brain. Therefore, in order to better annotate the genomic functions and to better understand relevant genetic diseases, about a decade ago, people invented the RNA sequencing technologies. The RNA-seq data are generated from the unobserved full-length RNA isoforms in RNA-seq experiments. And then, based on this data, we want to use statistical inference to recover the hidden RNA isoform information. In the next couple of slides, I want to briefly discuss the technical details of RNA-seq technologies because it's important for us to understand the data format. So please bear with me if you're already familiar with this technology. We measure the length of RNA sequences using the number of nucleic acids. For example, this short exon contains 6 nucleic acids, and we say it's 6 base pairs long. But in human bodies, the full-length RNA isoforms are more than 1,700 base pairs on average, much longer than the length the sequencing machine can handle. Therefore, the first step in RNA-seq experiments is to randomly break these full-length isoforms into shorter fragments. And then, after several processing steps, the sequencing machine will generate short sequences of fixed length from the 2 ends of each fragment. These short sequences are typically smaller than 300 base pairs, and we call them RNA-seq reads. Based on this generation process, it is straightforward to see that the number of RNA-seq reads coming from each isoform is proportional to the abundance and the length of each isoform. Once we get these RNA-seq reads, we can compare the nucleic acid sequences in the reads to the sequences in the genome to determine the starting genomic position of each pair of reads. And based on this information, we can summarize a histogram of RNA-seq read counts for the whole genome by counting the number of RNA-seq reads overlapping with each genomic position. Based on this mapped RNA-seq data, we can ask biological and statistical questions at different levels. For example, at the isoform or transcript level, we can ask what are the truly expressed isoforms in a given biological sample and what are their corresponding expression levels. At gene level, by quantifying the number of RNA-seq reads mapped to each gene in different biological conditions, we can study whether a gene's expression changes among different cell types. And finally, at the level of biological samples, we can compare multiple RNA-seq samples based on their transcriptomic similarity. And in the first part of this talk, I want to introduce 2 statistical methods developed to improve the transcript level analysis based on bulk RNA sequencing data. The first method I want to introduce is called AID, and it is developed to improve isoform discovery with high precision. As I have mentioned, this is a very critical question in many biomedical problems because more than 90% genes undergo alternative splicing in mammals, and at least 35% genetic diseases involve abnormal splicing. So it's very important for us to have an efficient tool to help us determine which isoforms are truly expressed in a given biological sample. And based on the mapped RNA-seq data, we want to build a model to infer which isoforms are truly expressed among all the candidate isoforms. This is a very challenging task due to 2 main reasons. The first challenge is that we have a very large variable set because the number of candidate isoforms increases exponentially with the number of exons in a gene. For this 4-exon gene, we already have 15 candidate isoforms, but for most human genes, we have much more than 10 exons. And among that large variable set, we need to identify a small set of truly expressed isoforms with high precision. The second challenge is the great information loss in RNA-seq experiments. Since most RNA-seq reads are very short compared to the full-length RNA isoforms, most of the reads cannot be uniquely mapped to a single isoform. For example, if we look at this pair of reads, we know that it can come from these 4 isoforms, but we do not get to observe its true origin. and the probability that this weight coming from each isoform needs to be estimated by statistical methods. Finally, there are different sources of technical biases that are introduced into the RNA-seq data that further complicate our analysis. Since the invention of RNA-seq technologies, many methods have been developed for isoform discovery, and among them, the most popular methods, cuff-links and string type, are both based on graph-based models developed by computer scientists. And these two models do not take statistical uncertainty into consideration. Therefore, they may still suffer from low accuracy for genes with complex splicing structures. And even when some elements such as exons can be correctly identified, it is very difficult to correctly assemble them into full-length isoforms. And more importantly, the usage of transcript annotations in existing methods always result in a lot of false positives. The transcript annotations are databases with isoforms which are experimentally validated. For example, the Ensemble database contains more than 200,000 isoforms annotated for human genes. And incorporating these annotated isoforms in our models can help us increase the recall rate of isoform discovery. However, on the other hand, since the annotation set is constructed regardless of cell types, if we compared annotated isoforms with expressed isoforms in a specific cell type, the annotation set may not be very accurate. For example, if we compare the annotated isoforms with expressed isoforms in the normal brain, or the expressed isoforms in the brain of Alzheimer's or the brain of the Parkinson's, the annotation set may not be very accurate. And in existing methods, incorporating these annotated isoforms always introduce a lot of false positives. And this is not acceptable in biomedical research because too many false discoveries will make it very challenging for us to perform experimental validation and to identify the correct drug targets. Therefore, motivated by the need to improve the precision of isoform discovery, we developed the AID method to selectively leverage annotation information and therefore increase the precision and robustness of isoform discovery. The AID method will first construct a probabilistic model to account for different sources of technical biases. And then, based on this model, we try to conservatively identify isoforms that make statistically significant contributions to explaining the observed RNA-seq data. And I want to emphasize that AID is the first method that tries to control false discoveries in isoform discovery using a statistical testing procedure. So if we think of the isoform discovery problem, we have both the observed RNA-seq reads, which contain noises, and the prior annotation information, which is not 100% accurate. And by selectively using information from both sources, the AID model tries to identify a small set of isoforms with high precision. And in order to achieve this goal, we designed the stepwise selection procedure in AID to give priority to those annotated isoforms. And this stepwise selection procedure can be divided into two stages. And let me introduce the procedure using this 4-exon gene as an example. We suppose that this gene has 3 annotated isoforms, and the remaining candidate isoforms are not in the annotation set. Then, in the first stage, we only consider the annotated isoforms in our likelihood model, and the fractions of the remaining isoforms are directly set to 0. Then after initialization, we take forward steps and backward steps in turns to update our identified variable set. Each forward step aims to identify a new annotated isoform that has significant contribution to the likelihood model. And each backward step aims to remove one annotated isoform from the identified variable set to rectify the results, since an isoform that is initially significant may become trivial at a later step. So we first initialize using the annotated isoform that overlaps with the most number of reads. And then, in the forward step, we consider adding one annotated isoform into our variable set to improve the likelihood. This isoform is selected based on maximum likelihood estimation. And then, we compare this proposed new model with the current model using the likelihood ratio test. If the test result is significant, then we will move forward, and our current variable set contains 2 annotated isoforms. Then similarly, in the backward step, we consider removing one annotated isoform from this set to rectify the results. And again, this isoform is selected based on MLE, and we compare the two models using the likelihood ratio test to determine whether we should remove the isoform or keep it. And we will repeat this process until the model determines that we can no longer move forward. Then we will stop and move on to the second stage, in which we start considering all the possible isoforms in addition to the annotated ones. And again, we take forward and backward steps in turns until the model converges. Using this stepwise selection procedure, we can effectively give priority to those annotated isoforms while ensuring that only isoforms that have statistically significant contributions to explaining the RNA-seq reads will be included in our final variable set. And in order to introduce the likelihood model of AIDS in detail, let's consider a gene with M exons. And we suppose that after the mapping or alignment step, we have n RNA-seq reads mapped to this gene. Then in the problem of isoform discovery, our observed data are RNA-seq reads, which can be denoted as R1 to Rn. The hidden data are isoformal regions of the reads, which are denoted as Z1 to Zn, and Zi equals to J if read Ri comes from isoform J. But we do not get to observe this information from the RNA-seq data. And our parameters of interest are isoform fractions, which are denoted as alpha J's, and they sum up to 1. In the problem of isoform discovery, our main goal is to identify the truly expressed isoforms whose isoform fraction are greater than 0. And based on this setup, we can derive the joint probability of one read Ri and its isoformal region Zi. This probability reduces to the product of Hij and isoform fraction J if read Ri comes from isoform J. Here, we use Hij to describe the generating mechanism of RNA-seq reads, and its formal definition is the probability of observing a read equivalent to Ri when we assume the read comes from isoform J. And I will talk about the estimation of Hij's in the next couple of slides. But now, based on this result, we can derive the joint probability of all the reads and their isoformal regions. And this leads to the complete log likelihood of the isoform fraction alpha. And based on this, the problem of isoform discovery is reduced to a constraint optimization problem, in which we aim to maximize the observed log likelihood subject to the constraints that the alpha j's should be greater than 0 and sum up to 1. It turns out that this problem can be solved using this EM algorithm. And in order to calculate the estimated alpha j in each iteration, we need to know the value of Hij's. Recall that we use Hij to represent the read generating mechanism. And if we think of one pair of read, it's actually characterized by two independent features: the starting genomic position of this pair of reads and the corresponding fragment length of this read, assuming that it comes from isoform J. Therefore, we calculate Hij as the product of two probabilities, PS for the starting position and PF for the fragment length. First of all, for the fragment length, we model it using a truncated log normal distribution based on our empirical observation and the fact that the RNA segments that are too long or too short are filtered out in RNA-seq experiments. For the starting positions, instead of using a uniform assumption, we build a multivariate kernel regression model to estimate different technical biases that may depend on RNA sequences, genomic positions, and exon lengths. And these technical biases make certain starting genomic positions more frequently sampled compared to the other positions. In order to train this model, we collect the RNA-seq reads that map to genes with a single isoform in the annotation, because the isoformal region are given in those cases. And with the trained model, we apply it to the remaining RNA-seq reads to calculate Hij's for every combination of indices I and J. And this completes the main component of the AIDS model. In interest of time, I will introduce one simulation study and one real data study to demonstrate the performance of AIDS. First of all, for the simulation study, we use the R package polyester to simulate RNA-seq data for protein-coding human genes with predetermined isoform fractions. And this package learns from real data to generate RNA-seq reads, so it's independent of our aid model, and the comparison is fair to odd methods. In order to evaluate the performance of isoform discovery given annotation sets of different qualities, I also designed 9 synthetic annotation sets with different precision and recall rate compared to the truly expressed isoforms. And these 2 box plots demonstrate the gene-level precision and recall rate of age and the other 3 state-of-the-art methods, couplings, string tie, and slide. And very apparently, we can see that aid outperforms these methods in terms of both precision and recall, especially when the annotation sets are not very precise. For example, if we look at the first 3 annotation sets, their overall precision rate is only 40%. But aid is able to improve the precision for a large number of genes because it implements the stepwise selection procedure and it will not be misled by the wrongly annotated isoforms. In addition to these gene-level results, we can also thresholding the estimated isoform expression levels at different cutoffs and get the overall precision recall curve of the 4 methods. And the red one corresponds to aid. And we can see that when all methods have the same recall rate, aid has the highest precision for all annotation sets. And the second best method, the blue one, corresponds to couplings. Since both aid and couplings have better performance in this simulation study, we applied these 2 methods to breast cancer RNA-seq datasets. And after we get the isoform discovery result, we randomly selected 6 genes that have isoforms identified only by couplings but not by aid, and we performed experimental validation using a technology called PCR. Our experimental validation results show that for 4 out of the 6 genes, the isoforms uniquely predicted by couplings were false positives. For example, if we look at the second isoform of each gene, it's only predicted by couplings, but the PCR experiments show that this isoform is actually not expressed in breast cancer samples. On the other hand, we also looked at the isoforms that were only predicted by aid, but not by couplings. And this displayed isoform is only predicted by aid and validated by PCR experiment for the gene FGFR1. Then we selected 2 breast cancer cell lines, suppressed the expression of this isoform in these 2 cell lines, and compare with the control. And in these figures, the intensity of the purple color demonstrates the number of cancer cells. And very apparently, we can see that the growth of the cancer cells was inhibited after we suppressed the expression of this isoform, showing that it may have function in the growth of breast cancer cells. And following up this experiment, we also did experiments to show that the function is specific to this isoform compared to the other expressed isoforms of the same gene. So to briefly summarize the aid method, it is the first isoform discovery method that tries to control false discoveries by implementing a statistical testing procedure. And by selectively using information from the RNA-seq reads and the prior annotation set, the aid model is able to identify a small set of isoforms with high precision. And this method is implemented as an R package, which is freely available on GitHub. So now if we think of the main purpose of the aid model, it aims to identify a set of expressed isoforms based on a single RNA-seq sample. And with the isoform structures predicted by computational methods such as aid, or with the isoform structures extracted from the existing annotation sets, a follow-up question is, can we improve the estimation of isoform expression levels if we have multiple RNA-seq samples coming from the same tissue or cell type? And the second method I want to introduce is developed to answer this question. This method is called MIS-IQ, and it is developed to jointly model multiple RNA-seq samples coming from the same biological condition. We were motivated by the fact that the RNA-seq datasets are accumulated at a very fast rate in public repositories. For example, the GTEx database contains more than 9,000 samples coming from 54 normal tissues. And the TCGA database contains more than 10,000 RNA-seq samples coming from 33 cancer types. So it would be great if we can have a tool to help us integrate the information from multiple RNA-seq samples. And more formally speaking, our question is, given DNA-seq samples and known isoform structures, can we estimate the abundance of each isoform for every gene? If we summarize the existing approaches to this task, they fall into two categories. The first category applies a single sample method to each sample separately and then take the average as the estimated isoform expression level. The second category applies a single sample method to all the pooled reads from the D samples. However, neither approach is able to consider the possible heterogeneity in RNA-seq datasets, and their estimation can be easily biased by samples with relatively poor qualities. For example, here I'm plotting the histogram of RNA-seq read counts mapped to the gene TPR in 6 human embryonic stem cell samples. And if we look at these gray shaded areas, for example, I can zoom in to the fourth one. These samples do not agree well with each other in terms of RNA-seq read distribution, even though they come from the same cell type. This shows that RNA-seq data can be highly heterogeneous, and we must take this factor into consideration in our modeling process. Therefore, in order to deal with this problem, we designed the MIS-IQ method. And given multiple RNA-seq samples coming from the same biological condition or the same cell type, the MIS-IQ model first tries to identify a consistent group of samples with homogeneous and better quality, and then we want to improve the estimation accuracy of isoform expression levels by giving higher weights on this consistent group. And in order to introduce the MIS-IQ model, let me quickly review the notations for a single RNA-seq sample. Suppose we consider a gene with J known isoforms. Our observed data on RNA-seq reads, which are denoted as R1 to Rn. The isoformal regions of these reads are denoted as Z1 to Zn, and the isoform fractions are denoted as alpha J's, and they sum up to 1. Now suppose we have D RNA-seq samples coming from the same cell type, then we can introduce a superscript D in the notations of R and Z to represent the RNA-seq reads and isoformal regions in sample D. In addition, we introduce a second type of hidden data ED to represent the sample consistency indicators. If sample D belongs to the consistent group which has homogeneous and better quality, then ED equals to 1. Otherwise, ED equals to 0. And this time, our parameters of interest are still the isoform fractions. But our main goal is to estimate alpha, which is the common fractions of samples in the consistent group. While for sample Ds that do not belong to the consistent group, they can have their own isoform fraction beta D, which is different from alpha. And to unify and simplify the notations, we use tau D to represent the isoform fraction in sample D. Based on this setup, we build a hierarchical model to explain the generation of RNA-seq reads in multiple RNA-seq samples. First of all, we'll assume that the sample consistency ED follows a Bernoulli distribution in which the consistency proportion gamma has a beta prime. And then in each sample D, its isoform fraction tau D either equals to the common fraction alpha or the sample-specific fraction beta D, depending on whether sample D belongs to the consistent group or not. And both alpha and beta D are assumed Dirichlet prime. Finally, in each sample D, we assume that the observed RNA-seq reads are generated from the hidden full-length RNA isoforms based on the sample-specific isoform fraction tau D. Given this hierarchical model, we can derive the joint probability of all the reads, isoform fractions, and other hidden variables. And this probability can be decomposed into 4 components, each representing the probability of observed reads or hidden variables. This is a very complex probability distribution whose direct estimation is not feasible. Therefore, we designed a deep sampling approach to draw posterior samples from this distribution. And In order to reduce the computational burden, we first integrate out tau from this distribution based on the property of Dirichlet distributions, because the inference of tau is straightforward once we know the values of isoformal regions Z and sample consistency E. And then we designed the Gibbs sampler to draw posterior samples of Z, E, and gamma based on their conditional distributions. Here, we use the capital T to represent the total number of iterations available after the burn-in period. And finally, our MIS-IQ estimator is defined as the average of the estimated isoform fraction in each iteration. And we can look at the derived formula for isoform fraction J in a single iteration, and it can be viewed as a weighted average of the prior information lambda in the Dirichlet distribution and the observed RNA-seq information in the consistent group when EDT equals to 1. With this MIS-IQ estimator, I first want to use two examples to demonstrate why it is necessary to consider the heterogeneity in RNA-seq datasets. And I applied the MIS-IQ method to 6 human embryonic stem cell RNA-seq samples. And in these two figures, the numbers demonstrate the isoform indices. The y-axis is their corresponding isoform fractions, and the x-axis denotes the 6 samples. If a sample is identified as in the consistent group by MIS-IQ, then it's labeled as yes. Otherwise, it's labeled as no. And this first example gene has 6 isoforms, and 4 out of the 6 samples are identified as in the consistent group. This second gene has 12 isoforms, and 3 out of the 6 samples are identified as in the consistent group. If we look at samples in the consistent group, they have very similar estimated isoform fractions. And this shows that if we allow higher weights on this consistent group, we have a better chance to improve the quantification of isoform expression levels. We also compare the performance of MIS-IQ with several other quantification methods based on human genes with multiple annotated isoforms. And in each experiment, I make 6 RNA-seq samples in the consistent group with 3 other samples and calculate the error rate of each method after isoform quantification. And in these figures, the first box plot demonstrates the error rate of MIS-IQ. We can see that it achieves much smaller error rate compared to the remaining methods, especially when compared to the simple averaging or polling method denoted in orange and purple colors. As a summary of the MIS-IQ method, this is the first isoform quantification method that tries to consider the possible heterogeneity in RNA-seq datasets. And by modeling the sample consistency, MIS-IQ can support better evaluation and reuse of public RNA-seq datasets. And it's also implemented as an R package for public application. And this concludes the first part of this talk.
So I have one question. So
Sure.
when you weight the samples, that's for all the genes together? Or you do it the gene by gene?
We do it gene by gene.
Okay.
And for the second part of this talk, I want to discuss statistical methods for single-cell RNA-seq data. And in single-cell RNA-seq experiments, the RNA molecules are taken from isolated individual cells. But we can label each RNA transcript so that after the sequencing step, we can tell which cell it comes from. So for each individual cell, after the mapping or alignment step, we can summarize its RNA-seq profile. And first of all, I want to introduce a statistical method called S-impute for the imputation of single-cell gene expression values. And then, very briefly, I will talk about the tool we developed to improve the experimental design of single-cell studies. If we think of the traditional bulk RNA-seq technologies, the RNA molecules are taken from a group of cells. So if we want to quantify the gene activities, we can summarize a gene expression vector by counting the total number of RNA-seq reads mapped to each individual gene. But with the emerging single-cell RNA-seq technologies, we're able to summarize a gene expression matrix in which rows represent different genes and columns represent individual cells. However, because of the technical limitations, there is substantial missing data in this gene expression matrix. there are usually many genes that are expressed but not detected by the single-cell RNA-seq experiments. And these genes will have false zero or false low count in this read count matrix. So we developed the S-impute method to identify and impute these missing values to improve downstream analysis.
So why the single cell has higher chance of missing values
It's
compared to the bulk
because
RNA?
the total amount of RNA molecules in individual cells are very limited. So it's
So
better.
they amplify the cell as well, right? Together, right?
Right, even though they do amplification, it's still very difficult to detect.
So you say
them.
they tag each cell so they can differentiate the rate? Okay.
Right. So in single-cell studies, the missing values are referred to as dropout values. And these dropout events occur when a gene's transcripts are actually expressed but not get sequenced in the experiments. And for example, if we randomly select two cells from the same cell type and observe their gene expression values, we'll observe exact zeros. And usually, we'll have many genes that have moderate to high read count in one cell, but have exactly zero count in the other cell. And these exact zeros complicate the analysis of single-cell data. And many statistical and computational methods developed for bulk data do not have comparable performance with single-cell data because of the sparsity problem. To deal with this dropout issue, we developed the S-impute method, and its main idea is that, first of all, for each gene, we want to determine which expression values are affected by the dropout events. And with this information, for each cell J, we can divide these genes into two subsets. Gene set AJ representing the genes that are affected by dropout events and are in need of imputation, and gene set BJ that are accurately measured with high confidence and do not need imputation. And next, for each cell, we want to impute the highly likely dropout values by borrowing information from the same gene's expression in similar cells. Let's still take this cell J as an example. Based on gene set BJ, we want to select the similar cells of this cell J, and then we want to impute the gene expression values of gene set AJ by borrowing information from these similar cells. And these main ideas are motivated by two important observations in real single-cell RNA-seq data. As an example, I'm plotting the gene expression distribution of 3 example genes in 3 cell subpopulations. And first of all, we can see that all this distribution has a bimodal feature, in which the first mode largely represents the dropout values, and the second mode represents the gene expression values that are accurately measured with high confidence. The second observation is that if we look at the same gene's expression distribution across different conditions, they are not the same. So in the imputation step, we must take cell subpopulations into consideration. And we designed the S-impute method to take a normalized and log transformed gene expression matrix X as input, and eventually it will output an imputed gene expression matrix with the same dimensions. The first step of S-impute is to detect the cell subpopulations and outlier cells. And since there are exact zeros in the raw count matrix, we apply the spectral clustering algorithm to principal components from PCA to obtain an initial clustering result. And based on this clustering result, for each cell J, we define the cells that are in the same cluster as cell J as its candidate neighbor set and denote it as NJ. And we will use this information in the imputation step. Meanwhile, for those cells that do not have any close neighbors, we define them as outlier cells, and these cells are excluded from the imputation step. The second step of S-impute is to identify the dropout values. Recall that the gene expression has a bimodal feature, so we model the expression of gene I in cell population K as a random variable coming from a mixture model. And we assume that with probability lambda I, it comes from a gamma distribution, which is used to model the dropout component. And with probability 1 minus lambda I, it comes from a normal distribution, which is used to model the gene expression values that are accurately measured with high confidence. And this lambda I is the dropout rate of gene I in cell population K. We can estimate the parameters in this mixture model using the EM algorithm. And with the estimate, we can calculate the dropout probability of gene I in each individual cell J. And this dropout probability is the probability that each gene coming from the dropout component in a cell subpopulation. And I want to clarify that this step involves two different concepts. First of all, each gene I has an estimated overall dropout rate lambda ik, and this does not depend on individual cells. It measures the overall dropout frequency of a gene in this subpopulation K. Second, every gene also has estimated dropout probability, DIJs, which may depend on individual cells. And this plot compares the observed gene expression distribution represented by the blue histograms and the fitted distribution given by the mixture model represented by black lines. With the estimates from the mixture models, we can identify the dropout values by setting a threshold on dropout probabilities. For each cell J, we define a gene set AJ containing all the genes with dropout probability greater than the threshold. And we think these genes are in need of imputation. Meanwhile, we also define a gene set BJ containing all the genes with dropout probability smaller than threshold. And we think these genes are accurately measured with high confidence and do not need imputation. By default, this threshold is set to 0.5 in our method, but we conducted a sensitivity analysis to show that the imputation results is not sensitive to the selection of this threshold. And as an example, I'm plotting the calculated dropout probabilities in 4 cells from a real data set. And we can see that most of the values are close to either 0 or 1, and the selection of this threshold only affects a small number of genes. Now with these identified dropout values, the last step of S-impute is to impute the gene expression levels cell by cell. And recall that we identified a candidate neighbor set Nj for each cell J based on spectral clustering result. Then, in order to select the similar cells of cell J from this candidate neighbor set, we construct a non-negative least square regression model based on gene set Bj, since these genes are accurately measured in cell J. And then, with the regression results, the cells that have estimated coefficients greater than 0 are considered as the similar cells of this cell J. With these estimated coefficients, we can predict the gene expression levels for genes in set AJ, and these predicted values are treated as the imputed values and will be output by S-impute. And I want to use 2 real data examples to show that these imputed gene expression values can improve downstream analysis. With the first example, I want to show that S-impute can help us identify cell clusters in real data. And in this example, I collected the gene expression data of 4,500 blood cells, and the proportion of 0 in the raw read count matrix is over 90%. And here, I'm plotting the individual cells based on the first 2 components from the Tisney algorithm. And these cells are colored by their actual immune cell types. We can see that based on raw data, different subtypes of T cells are grouped together and cannot be distinguished. But after we apply S-impute, the algorithm is able to divide each immune cell type into a distinct cluster. This shows that the imputed data better capture the cell identities in this example. And for the second example, I want to show that the temporal dynamics of single-cell gene expression can be recovered by S-impute. And in this example, I collected both bulk and single-cell time-course RNA-seq data profiled at different time points during the differentiation from embryonic stem cells to endoderm cells. And we can take a look at this gene, for example. In this plot, these black triangles mark the gene expression values calculated based on bulk data, and the box plots demonstrate the gene expression values in single-cell data. Based on the expression values from bulk data, we can see that the mean expression of this gene increases along the differentiation trajectory, but this dynamics is not revealed by the single-cell data because of the dropout events. And after we apply S-impute, we're able to observe a consistent pattern of increasing mean expression through the single-cell data. Similarly, for this second gene, the bulk data shows that its mean expression first decreases and then increases along the differentiation trajectory. But still, only the imputed single-cell data is able to reveal this temporal dynamics. And to quickly summarize, the S-impute method is developed to address the dropout events prevalent in single-cell RNA-seq studies, and its main advantage is that it only aims to identify the dropout genes and impute their gene expression levels, while trying to retaining the gene expression values that are not affected by the dropout events. And lastly, I want to very briefly introduce the SC design method, which is developed to improve the experimental design of single-cell studies. In the sequencing experiments, given a fixed budget, which means a limited sequencing capacity, we need to make choices between sequencing more cells with shallower sequencing depths in each cell, or sequencing fewer cells with deeper sequencing depths in each cell. And since 2009, more than 20 protocols have been developed for single-cell RNA-seq experiments. These protocols are able to handle different number of cells, and their resulting gene expression data have different levels of sparsity. So here, the main trade-off is that given the same budget, which means a fixed sequencing capacity, if we want to sequence more cells, then the resulting data will have more dropouts and tend to be more sparse. However, there is currently no unified criteria to help us determine the optimal cell number in single-cell studies. So we developed the SC design method to help researchers make a more objective choice. And the SC design method is able to learn both gene-wise and cell-wise parameters from both real single-cell RNA-seq data and the mixture model proposed by S-impute. And based on these gene-wise and cell-wise parameters, it's able to simulate synthetic RNA-seq data that can well capture the characteristics in real data. Then, based on these synthetic data sets, we can evaluate the choice of cell numbers from the perspective of detecting differentially expressed genes between two biological conditions. As a specific example, suppose we want to detect the differentially expressed genes between two types of glial cells, astrocytes and oligodendrocytes. Then we can apply the SC design tool to simulate RNA-seq datasets with a fixed sequencing depth but varying cell numbers. And then, based on each pair of RNA-seq datasets with varying cell numbers, we can evaluate the accuracy of differential expression analysis based on different criteria such as precision, recall, or F scores. And for each criterion, we can report the optimal cell number of the two cell types. Based on these results, researchers can guide their practical single-cell RNA-seq experiments and according to their specific research questions. And this method is also implemented as an R package, which is available on GitHub. So to conclude this talk, in the first part of this talk, I introduced two statistical methods, A and MIS-IQ, to improve the isoform discovery and quantification from bulk RNA-seq datasets. And by applying these two methods to RNA-seq datasets, we have a better chance to understand the alternative splicing process in different biological samples. In the second part of this talk, I introduced S-impute and SC design to improve the analysis and experimental design of single-cell studies. And as more and more sequencing data are generated, it is becoming increasingly important for us to develop powerful and interpretable statistical methods in order to uncover the hidden information from large-scale genomic datasets and to reduce false discoveries. As further data are collected, we're also better equipped to make advances in statistical methods to address emerging biological and biomedical questions. And finally, but not least, I want to add that the work I just presented today would not have been possible without the support and assistance I received at UCLA. And especially, I want to thank my PhD advisor, Professor Jessica Lee, for leading me to the field of statistical genomics, and for her support and patient guidance throughout my doctoral studies. And I also want to thank everybody in Jessica's group. And in addition to that, I also want to thank Professor Wu, Professor Zhou, and Professor Hoffman for serving on my doctoral committee and for their advice on my research and career development. I'm also extremely grateful to all my collaborators for their time and efforts on our research and for their great advice and suggestions. And throughout my doctoral studies, I also received numerous help and assistance from faculty and students in the Department of Statistics. And my sincere thanks also go to my family and friends, especially my parents, for their endless support. And I want to thank you all for coming to my defense, and I'd like to take questions. Thank you.
So we will let the general audience ask questions first.
Yeah, for this single cell part, you have the density of the log of the read counts. Like, how many samples did you use to draw that plot?
So you're referring to these examples?
Yeah, so the density of the read counts across all the samples, right?
Right, so in this example, we have about 200 single cells from the, I think it's from the mouse embryo data.
Okay.
So each histogram is based on a sample size of 200.
Yeah, on average 200 cells.
So all the three genes are from the same cell, right? No, from the same type of cell, and you have about 200.
So I have 3 genes, and I have 3 different subpopulations. And in each subpopulation, I have around 200 cells.
So later you showed some difference between single cell plot versus the mixed bulk cell, right? When you show that the imputation helps. Yeah, can you explain a little more? I wasn't able to follow. It's just such as that, right? So what do you mean by before?
Right, so I mean, so these black triangles demonstrate the gene expression calculated from bulk data.
That's measured on different time points.
Yeah, that's
right.
measured on different time points.
Now how about the single cell RNA?
Single cell is the same. So at each time point, we measure the gene expression in multiple individual cells. So
right.
Those are demonstrated by box plot. And
okay, so under the let's say look at the, for example, 72 hours, right? You have the old technique, right? The bulk measure, that's triangle. Then you have the same cell. You now measure the single cell RNA-seq, right? Then you get the distribution.
Right, so basically what they did is that initially they have the biological samples,
right.
And they divide the biological samples into 2 parts. For one part, they conducted the bulk RNA-seq experiment, and that gives a single value for each gene. And for the second part, they performed the single cell RNA-seq experiments, and for each gene, they get the gene expression values in the individual cells.
Yeah, so that's the box part, right?
Right.
It's the distribution across the different single cells, right?
Right.
Under the same biological sample, right? And then you clean up, you get that distribution, right? So then from left plot, so what would be the advantage of the single cell, right? You just didn't even measure better than the bulk RNA, right? That was my question.
Right.
You know, this is like a counterexample for single cell data.
Right. The main reason for doing single cell RNA-seq is that with bulk data, we only get this mean expression value. But if people are interested in the variance and in the distribution of gene expression among the cell population, they need to rely on single cell technologies.
So that has not been. That's not illustrated by this example, right?
I think many people do single cell RNA-seq simply because now they have the technology. They can claim, oh, we can measure individual cells while we cannot before. So that's one thing. Another thing is that they want to identify some unknown cell subtypes from single cell data. That's another reason why they do this. But they are aware that the data quality is an issue. So that's why we developed the imputation method.
Right, and it also depends on the individual genes. For genes that are highly expressed, single cell experiments may reveal some interesting findings. But for lowly expressed genes, because the sensitivity is not highly enough, then we need the assistance of statistical methods to help us improve the data quality.
So for the single cell data, do you see a lot of heterogeneity among the cells?
Yes, definitely.
If that's too much, then your imputation will be more challenging, right? You rely on the cell similarity so you can impute the value, right? Otherwise, if in the extreme case all the cells are, in fact, let's say under this time point, they're actually from different internal time points, right? So it's even questionable how you should group them in that case, right?
Right, it definitely depends on how we can perform in our first step when we try to identify the cell populations. And as I mentioned, there are many different protocols developed for single-cell RNA-seq experiments, and the imputation performance also differ from protocol to protocol.
So imputation is like the pretty standard first step to do the single-cell analysis?
I think it's controversial. Some people want to try it.
Hmm.
It's really, I think, from data to data. Because some people, if they think, oh, if I observe my data, I think there are definitely a lot of zeros that should be corrected. They will try imputation to see if that helps. But if they think the data looks fine, they may just proceed without imputation. So I would say in the single cell field, it's very subject to the researcher's knowledge about the cell they study. Otherwise, they cannot totally just rely on the data to make conclusion because the data is just too noisy. Yeah. So hopefully, but the field is moving very fast. Unlike the bulk RNA-seq data, for which Illumina has totally dominated the field, right? Everybody's using Illumina protocol. Single cell is still like a battlefield. A lot of protocols are still competing with each other, and it's very subject to the lab's choice of which protocol. So I feel like it's not unified yet. Yeah. Do we have other questions? I think there used to be like a hand right here.
Just a quick clarifying question. For the non-negative least squares problem where you find neighbors,
Yes.
do you also have a penalty term, or is it?
Oh.
Because you say you choose neighbors such that the coefficient is greater.
Yeah, that's a great question. So initially, we actually tried lasso, and then it turns out that just by using this non-negative least square regression, it also has an effect to the variable selection. And we compared the performance, lasso is not better than this one, so we just stick with this.
And another advantage is you want to have non-negative coefficients, right? That's what you want.
Also, in the isoform your methods, do you, when you compute those probability of different isoforms, do you consider all possible isoforms, no matter it's annotated or not? Is that the case?
Yeah, so we divide our method into two stages. In the first stage, we only consider the annotated isoforms.
But eventually you consider all possibilities,
Yeah,
right? So that can be a lot, right?
That can be a lot, yes.
So do you think your method is robust to that? Because that can be solid, right? I would.
So what we did in practice is that if a gene has more than 25 exons, then we only consider the isoforms whose exon-exon junction is supported by at least one ring. So if there's a junction that is not supported by any ring, then we won't consider that possibility, and that helps us reduce the candidate number.
It's like you did the screening. Yeah.
Also, for your GIP sampler, do you see does that actually converge in this big model?
I think it depends on the genes and the RNA-seq data.
That's also you need to consider all the isoforms, right? Except for the isoform problem.
For that one, no.
For that one, no. But for some genes,
they're only annotated as isoforms.
Right, but for some complex genes, even the number of annotated isoforms is large.
So basically, we recommend using the two methods in her part one. in a sequential way. So first try 8 to discover new isoforms, add them to the annotation set, and then do
okay.
extension.
Then restrict to the
Yeah, yeah.
So if you change give sample to original, it will be faster.
It will be,
Yeah, that could be an extension. Yeah,
Because it will take a
currently,
long time for keep sample to converge.
right. Currently, I haven't tried sample size larger than 10. So basically, we only consider like 10 RNA-seq samples coming from the same condition. But if we want to apply the method to large database such as GTAX or TCGA, then probably we need better computational efficiency.
And also you use gamma distribution for this dropout. So that
Right.
means the number of rates when a gene is actually dropout. What's the basis for this distribution?
So this is not based on the dropout mechanism. So basically, we try different distribution and find that this one gives us better model fitting. So in the field, it's also very popular to use negative binomial or zero inflated negative binomial on raw rate count instead of using the normal and gamma distribution on log transformed values. But we compare the data fitting and think our model gives better fitting.
So you think a mixture for gamma and normal can fit the observed data.
Right.
But that's a log transformed value.
Right, it's
Yeah,
So-
log transformed values.
Yeah.
Then why do you use gamma? Gamma is all positive, right? Why don't you just use 2 mixture normal?
When we do log transformation, we add a pseudo count of 1. So
Yeah.
the value will be positive.
Yeah, let you use your gamma, I will embed it into 2 gamma, right? Because they are both positive real line. But it's a little bit weird. Use 1 gamma and 1 normal, right? Their range don't even
Right.
overlap, right?
But it's mainly because it's very popular to model log transform gene expression values using normal distribution.
Yeah, I understand. In upper log transform, you can let's say you could just consider just 2 normal, right?
Mhm.
That would be fine. But gamma has a restricted support, right?
Do you feel like did you try like 2 normal before?
I think I tried 1 gamma and 2 normal. And in that case, sometimes the distributions can overlap, and we won't be able to identify dropout values because of the overlap. But I didn't try 2 normal distributions.
Because we want to capture the spike around 0. Since she added the pseudo count, so there will be a lot of 0.
Right, very
Maybe
small value, right?
Yeah.
After take log, it's very negative value, right? Or you
add
add
one.
1 to the
Add 1,
count,
yeah.
and 1.
So
So
the
zero.
theory is that your gamma distribution is only used to decide whether you need to impute.
That's right. That's
But
right.
can you find a more direct decision rule instead of fitting mixture model? I mean, fitting mixture model using EM
Yeah.
seems to be overkill for just making a decision whether you want to impute.
So the idea is that when we only observe a 1 mode distribution, even if the mode is close to 0, then we think that suggests maybe there's no dropout. So we just do not impute for that gene, right? That's the intuition behind it. So basically, she wants to first decide whether the fitting is better as a 1 mode distribution or a mixture model. If we choose the mixture model, then she wants to decide which values need imputation. So that's the whole decision-making rule. But if we want to drop the mixture model, we need to think about another way.
And also, when you impute, you want to find the neighboring cells.
Yeah.
But that can be also very tricky.
Right,
the
because
cells that you believe belong to the same type. That depends on your classing.
Right, it depends on the clustering and and the quality of the raw data. And initially, we also considered to do the clustering and imputation iteratively. But we find out that by doing it that way, we probably were over smoothing the data. So that's why currently we're just relying on the clustering results based on the principal
Which means you
component.
first perform PCA,
Right.
and then doing this spatial clustering.
Right. We're hoping that by doing PCA, it can reduce the negative effect of dropout values.
And another thing is that your number of clusters is a tuning parameter because that will determine how much you impute.
Right.
Yeah. Right. If you set it to be a large number, then you're basically imputing less. You borrow less information from the viewers.
So when you borrow information, for example, that one, the n sub j is a neighboring cell.
Right. So this
Oh,
is the
a
neighboring
number.
cell defined by the clustering result.
Oh, okay. So you mean these all the cells in that cluster
Right.
belong to this n sub j.
Right.
Can you just use some nearest neighbor? So you define metric and then find maybe say 100 cells that have closest to.
Yeah, I think we can also try that. So basically, after we identify the highly confident gene set Bj, we use this set to calculate the distance between every pair of each cell and identify nearest neighbors based on that distance. Yeah, but I cannot imagine like which approach would perform better.
And actually, we're very lucky that her method was the first one that got published in this field. Because before we started working on this, we just realized there's no such method in the field. So we just rushed out. And so that paper becomes the first one. But after we publish ours, I think there are like 40 papers of different imputation methods doing the same task. So they tried different approaches. I believe there's one paper that used the nearest neighbor approach. And there's another paper, many papers try to do just matrix completion. Yeah.
Matrix completion, but does that make sense?
Yeah.
Non-native matrix factorization kind of thing.
That's right. Yeah, and also another thing is that a lot of methods that use matrix factorization to do the completion, they just regard all the zero values as missing value. Right. But we think that's not right because a lot of the zeros are true zeros. So that's why I think the first step is critical. But after we identify the values that need imputation, then how to impute them? That's the second step. This one is subject to many choices. Right.
I think we actually tried to replace this step with matrix completion.
We tried that.
The performance is not good.
So what's the rationale for matrix completion? What are the hidden factors?
They think I think for the papers who use matrix completion, their rationale is that the matrix should be low rank.
Why is that?
They think because there are a lot of cell subpopulation. So cells in the same population will have high correlation. So the matrix should be low rank instead of low rank.
Okay.
Yeah.
It's like here, even though we have many cells, we only have a limited cell subtype.
Yeah.
And for this multiple, what is called MSIQ.
Yeah.
So that's essentially an outlier detection mechanism.
Yeah, that's another way to interpret the problem.
Yeah, it's just that we're defining the outliers as a kind of hidden thing, right? We have to find an outlier based on the hidden isoform proportion.
So far, what's the most successful story in terms of single cell RNA-seq. In terms of biological discovery, did they use that to?
I think we're still waiting. I think it's
but
more.
The technology is there.
It's there. There's
not unified yet, but
right.
In terms of scientific discovery, they're still trying to.
Right. I think most papers are just data generation.
Okay.
They just generate data and publish the data, and that's it. Yeah. I think that's a critical question. Many people raise this question: what did you discover? I feel like a lot of the findings in those data papers were just confirming our knowledge. Yeah. Just trying to justify, okay, this confirmation suggests that the technology is working, more like that. Yeah.
But do you see the technology will keep improving?
I think so. Yeah, I think so. It's improving. And there's another direction which I'm not sure which one would win. So one is the sequencing direction. People try to sequence individual cells to measure gene expression. The other direction is the imaging. People just try to develop some good techniques to just directly capture the fluorescence of gene expression and use that. So right now,
single cell.
All single cell, yeah. So the imaging technique is better in accuracy, but it's limited by the number of genes you can detect in every cell. The other way, the sequencing one is good in terms of genome-wide, being genome-wide, but you have very bad accuracy. So,
do they have a way? Let's say if you want to define a subset of target genes by just sequencing these target genes in a single cell.
For now, they cannot.
They cannot.
They have to do it genome-wide. Yeah, yeah. But there are some technology or protocols by individual labs. For example, when they prepare the library before they do sequencing, they can design some probes to enrich the genes they are interested in. They can do that, but it's more like lab to lab. Yeah.
So over the past 3 to 4 years, people are devoted to modified protocols so that they can sequence more and more cells. Initially, they can only sequence hundreds. Now, some technologies can sequence thousands, even to millions. But with the dropout issue, I think now they're spending more time thinking about how to improve the data quality while sequence a large number of individual cells.
But you can sequence a large number of cells, maybe they can afford just to throw some of the cells. Right? If you find a lot of dropout, just throw them away.
And in the first part, you were able to recruit some biologists to do the wet lab experiment.
And for the that's our collaborators in China.
So you convinced them to do it for you.
Yeah, it's actually a long story. Yeah, but I met him actually when I was an undergraduate student, and he was a PhD student. But we knew each other back then, and then we started this conversation just randomly.
But
they're happy
it's hard, right? It's
usually it's hard, yeah. But the good thing is that they actually use our method 8, the first talk, to find some discoveries from breast cancer data. So they're very happy for that because they may be potential markers for diagnosis. Yeah.
Well, for the experiment validation, I have a minor comment.
Yeah.
You show that the one that identified by the other methods was a cufflink or what?
Yeah.
Cufflink has false positive. That's not a good criticism because most methods will find other false positives.
That's right. That's right. Yeah.
Right.
Yeah.
You test another method, maybe you can equally or even higher chance of false positives. It's better. The second part, when you verify yours, it's good. That's more like evidence.
That's true. It's just that we randomly picked some ones found by conflict, right, or found by us, and we do separate validation and found that there are more false positives in the cufflinks cohort. It's more like that.
Oh, the unique sets.
That's right. Yeah, yeah. I think that's an issue I realize more and more. It's like in the field of bioinformatics, many people are trying to argue more findings as better. But more findings may not necessarily be better. You have to verify how many of them are false positives, but they just didn't consider that, like couplings. Yeah.
And this library, this annotated isoform library, will keep improving.
Yes, yes, yeah. The problem is that for human, a lot of the annotated isoforms are only from blood because it's easier to get blood samples from people.
I see.
But for other tissues like brain, those things for which it can be very difficult to do biopsy, right? Especially from normal people. So that's why the annotation is still very incomplete.
So how large is this library?
So, on average, about 200,000 isoforms for 20,000 human genes.
I see. Yeah, in the end, I want to comment that you talk extremely clear. Even to lay audience like myself, I can understand most of your talk. When I go to other bioinformatics talk, it become very I got lost very easily.
Actually, she tried a lot of efforts to polish it because this is part of her job talk, actually.
Yeah, it's very polished.
Yeah.
And very clear to the statistical audience.
Okay, do we have other questions from other audience? If not, then we will actually close the door and we'll discuss for the committee. Yeah. So Alice joined. But he sent me an email.